Hi, everybody. We will wait for a couple of minutes for let everybody to join in, and then we will start. Uh, so I'll just be here for 5 minutes. We will wait and see if everybody can join in. Then we can start this. Hello, welcome to the fourth session. We will be starting in a few minutes. I'm just trying to connect my iPad with this. Uh, yeah, I hope the screen is visible. Welcome to the weekly live interactive sessions of the course Enzyme Science and Technology. We are in the session 4, week 4 of the 12 weeks that we have. And today's uh, week is mostly about overexpression of proteins. So let's look at initially what we will be doing is going through a brief summary of whatever has been taught this week, and then we will go with some question answers. So for protein expression, the very first step that needs to be done is the DNA delivery into the host. Now, the surface chemistry of the host cell plays a very important role. So essentially, they can either attract or repel DNA. Both can happen. Now, there is a cell wall and the membrane which causes which becomes a barrier for DNA to enter the cell. So we need to prepare cells which are called competent cells. So competent cells, um, they are they readily take up foreign DNA. In this case, it would be recombinant DNA, right? And the process by which this foreign DNA is inserted into the competent cell is called transformation. So I think so far is known by everybody. Now, there are several agents which make a cell competent. So the bacterial strains and their corresponding competent agents are discussed. So let's say Streptococcus pneumoniae, fine. It can be made competent using mitomycin C, fluoroquinolone. Similarly, for Bacillus subtilis, we have uveolite. Helicobacter pylori, we have ciprofloxacin. For E. coli, which is used in lab, we have calcium chloride or rubidium chloride, right? Things like that. So how to prepare competent cells rather. So the bacteria is usually incubated with divalent cations. So it could be like calcium chloride, magnesium chloride, rubidium chloride, something like that, for 30 minutes at 4 degrees, right? So the cell walls, they swell. DNA uptake in a bacteria is more at its growth phase, which is essentially the log phase. So whenever we try to prepare the competent cells, we keep the OD600 around zero point 0.6 to zero point 0.8, because this is the position, this is the growth at, this is the point at which the bacteria is in its log phase. Here it is more competent. So transformation is the process by which we insert this DNA into the competent cell. And after the DNA is inserted, we calculate transformation efficiency. Transformation efficiency is the number of colony forming units or also known as CFU by transforming, let's say, 1 microgram of plasmid. So here we get the transformation. There's there's a formula for getting transformation efficiency as well. And the unit of transformation efficiency is, as I already told you, CFU per microgram. That is the unit. So you can look at it, the slides, to understand how this works. Now coming to electroporation, it is another way of transformation. Again, here we give high electric pulse and it disrupts the cell membrane, allowing entry of DNA. Right? There are several factors that affect the transformation efficiency. One is the plasmid size. So if the plasmid size is higher, the transformation efficiency would be lower, definitely. Then the form of DNA which we of so let's say supercoiled DNA can easily get inside compared to linear DNA. Then talking about the genotype of cells. So like the cloning strains of E. coli are E. coli K12 strains. These are the preferred lab strains. So they have higher competence or transformation efficiency is also higher. Obviously, the growth phase, that is the log phase, is the best phase for highest transformation efficiency. Then we come to the method of transformation. So there could be like chemical method or electroporation that can also determine the transformation efficiency. And whether there is any damage to DNA, there could be UV damage, or anything else, that can also cause problems. Now, talking about DNA delivery in yeast and mammalian system. So, we already talked about bacteria, but what about yeast? So, we used, in case of yeast, using lithium acetate DNA and PEG. So this basically contains lithium acetate DNA. This DNA is not the DNA that you want to insert, but rather these are carrier DNAs. These are important because in the cell wall of yeast, there are some non-specific DNA binding sites. So your plasmid can go and bind to these sites and not enter the yeast cell. So to prevent that, these carrier DNAs actually block these sites and the transformation efficiency increases. In mammalian cells, I'm so sorry. Uh, DNA delivery could happen using chemical transfections methods. We'll talk about it. So chemical transfection, the one of the most preferred, I mean, not really preferred chemical transfection as such, but has been reported using calcium phosphate, very similar to calcium chloride in case of bacteria. Here we have chemical transfection agents, although they are not very efficient because transfecting eukaryotic cells, mammalian cells, is a little bit difficult. Talking about liposome or plexes, these liposome lipoplex methods are, they are polycationic carbohydrates. Polyamines, stuff like that. So lipofectamine is another in the example. Then we have bactofection, or basically using bacteria to deliver DNA. So the most, it's in case of Agrobacterium tumefaciens, which has TI plasmid for plant cells. And then there is transduction, where viruses are used. So for example, adenovirus or adeno-associated virus, these viruses are used for transfection. So if we talk about chemical transfection, we already talked about it. So calcium phosphate method is used. Um, Not very much like used. In case of liposome and lipoplex transfection method, um, so there is a lipofection reagent, which is basically a lipid. This is, let's say, a lipid, uh, mostly a lipid, and there is a plasmid DNA, let's say. They form a complex. It's a DNA lipid complex. And then it goes inside the cell. this complex goes inside the cell through a process called endocytosis. So this is how it works. Then we have bacteriophage, where bacteria is used. So for mammalian cells, some bacterial species are like Salmonella or Shigella can be used, but usually not very much preferred because then bacterial cells they outnumber the growth, or in case of in mammalian cells becomes a problem. Hello. Yes. Don't see anything actually. Don't know where the problem is. Okay, I can now I can see yeah, yeah. Uh so yeah. I'm actually using the same link. I don't know why this problem happened. Should I continue? Yeah. Okay. Yeah. Let's so yeah, as I was telling, for mammalian cells, Salmonella or Shigella, they are used. Um, so most bacterial cell strains are attenuated if they are used for um, transduction. So if we or for bacteriophage, transduction we use viral cells. So we used phages, bacteriophages. Then comes the screening. How do we screen the cells which are actually transformed? So we used enzyme. So enzymes can be used. For example, we can perform blue-white screening. We will talk about it. Uh, then there is the antibiotic resistance gene. Or the phenotypic changes. So talking about the enzyme, um, so there's a colorless compound which is called XGAL. This is the principle of blue-white screening, which is actually 5-bromo-4-chloro-3-indolyl beta D-galactoside. And the enzyme is beta galactosidase. So this is the enzyme, the substrate is XGAL, and this converts XGAL into a blue colored product. So this is the principle behind blue-white screening. Then there is antibiotic sensitivity. So there are multiple antibiotics. For example, ampicillin, kanamycin, tetracycline, chloramphenicol. Okay, the gene products are like beta lactamase, neomycin phosphotransferase, then there are ribosomal protection proteins. The mechanism is like degradation of ampicillin, kanamycin. Okay, anyway. Now, insertional inactivation is another method of screening. What happens here is the foreign gene is inserted into a functional gene in between a functional gene, so it becomes non-functional. So let's say if LAC-Z, which codes for beta galactosidase, the enzyme that we found out in case of blue-white screening, if we insert something in the LAC-Z cassette, there will be no functional beta galactosidase, and this is one of the methods of screening that can be performed. Now, another thing is complementation of mutation. So let's say in this case, the host cells are engineered. Fine. the host cells are engineered in a way where uh, the missing genes of plasmid, sorry, missing gene of the host is present in the plasmid encoding the gene. So that gene is there in a plasmid. So that if let's say if that gene is important for survival, only the positive clones will survive. So what there are certain requirements for this. So host strain should be deficient of the gene. Second, a defined media. And a vector containing gene to supply the gene product. Fine. So there could be positive or negative selection. So in case of positive selection, what happens is the host strain, it does not grow on the media lacking the functional gene, but host transform with the recombinant DNA, they can grow. Another one is negative selection. In this case, what happens? Chemical compound is added that is usually cytotoxic. So the gene present will break down this chemical so the genes which are transformed will grow, but the wild type cells, they will not grow because of the toxic compound, right? How to screen mammalian cells? So there is reporter gene assay. One of the most preferred one is, let's say, luciferase assay. What happens is there's a promoter and luciferase gene attached to it. So what happens is there's a substrate that will basically, this reporter gene will have an enzyme. It will convert this substrate into a product, and this product will either emit light or luminescence or anything. So that can be helpful here. So there are certain genes which are popular for, and their product we will see, and the reaction also. So the genes are, for example, uh, the gene CAT is chloramphenicol acetyl transferase. The reaction catalyzed is from chloramphenicol to acetyl chloramphenicol. Okay. Similarly, LAG-Z for it codes for beta galactosidase. We already know this. It the reaction is O-nitrophenol beta galactoside to O-nitrophenol and galactose, right? Similarly, Luc is for luciferase. It converts luciferin to oxyluciferin. Similarly, alkaline phosphatase is also there. It releases inorganic phosphate. Then GFP, green fluorescent protein. It gives fluorescence. So luciferase reported gene system, luciferase is taken from the abdomen of a firefly. Photinus spiralis is the scientific name, right? The substrate is D-luciferin, and the product is oxyluciferin. In this process, there is emission of light, and cofactors are ATP and magnesium. It's a very, very quick reaction. Hence, the readings need to be taken quickly, very fast. Otherwise, because this light emission happens for few seconds. So if we look at it, how it works. So luciferin converts to luciferin adenylate using ATP, right? So if ATP is so it's usually a kit-based assay. When done commercially, it's when this ATP is not present, might not work. And then there is decarboxylation reaction to form oxyluciferin. And it basically, this process emits light, or photons rather, to be more specific. So there could be GFP chimeric construct where a protein, let's say this is your protein X, is fused with a GFP, and this GFP will fluoresce. It has fluorescence, and you can view it in a microscope very easily. For localization of proteins, this kind of GFP chimeric constructs are created. Now, how to confirm a cloned DNA? They're the most preferred. DNA sequencing can be performed to check whether your cloned DNA is present. So there are two methods for this. One is the Sanger's method, which uses dideoxy nucleotides, DDNTPs to be specific. So what they do, it is also known as a chain termination method, where what happens is during a reaction, there will be addition of bases. But if there is a DDNTP, let's say DDATP is added at one point, the chain will terminate and it will not form amplification. So what happens is, in a gel, you would see multiple amplicons of different length in a gel, and those are actually sequenced because they are one base pair. If you go to a one base pair resolution, you can actually form the sequence. Another method is Maxim-Gilbert sequencing. So here there are base specific reaction. For example, dimethyl sulfate, it reacts for guanine. Similarly, formic acid, uh, it reacts with guanine and adenine. It cuts basically there. Then there is hydrazine, and which cuts with thymine and cytosine. And if you add NaCl to it, it only cuts at cytosine, right? Uh, so vectors can be of two types. Again, they can be either cloning vectors or expression vectors. In case of cloning vectors, you check for the mechanism of transcription, replication, things like that. I'll be just Yeah, there could be expression vectors as well. Um, okay, so cloning vectors and expression vectors, as I already told you, cloning vectors are to understand the mechanism of transcription, replication, genomic, and cDNA library preparation, something like that. We require cloning vectors. Expression vectors to understand the mechanism of translation. We require these vectors. And protein production. So for enzymes, we would require mostly expression vectors. Now, understanding how protein synthesis works, we need to understand that first, the RNA polymerase, it binds to the promoter. So when it binds to the promoter, there is start of transcription, which leads to formation of mRNAs, right? So after mRNA is formed, mRNA leads to protein synthesis, and this is usually coupled. So there are tRNAs which are involved, which reads the mRNA using anticodons, and then it works. Now coming to the host and vectors, so there could be multiple host cells which we can use. So they could be either prokaryotic cells like bacteria, they could be yeast, they could be animal cells, or even plant cells. And usually the transforming agent or the DNA that we are transforming is usually a plasmid. Mostly it's a plasmid, but there could be, like artificial chromosomes and stuff like that as well, which can be transformed. But usually for proteins, usually a plasmid is sufficient to get your gene of interest inserted. Now, criteria to select an expression system is to understand how much protein do you need. Quantity of desired protein, the size of the protein, compatibility, and the downstream application. Now, in an E. coli expression system, we have a promoter, which is the docking site for RNA polymerase. We have a ribosome binding site. Now, this ribosome binding site usually has Shine-Dalgarno sequence. This is the docking site for ribosome, obviously, as the name suggests. Then there is a termination site where mRNA is terminated. mRNA synthesis is terminated. And there could be affinity tags before or after the protein, right? Before or after the gene sequence. So there are multiple affinity tags. Very common are polyhistidine tags. There could be other tags as well. So uh, these are present. An IPTG inducible promoter is a promoter which can be induced in the presence of IPTG. So there are a number of promoters which are designed for overexpression only when IPTG is given. And as we already know, IPTG is a synthetic analog of lactose. lactose, and it can essentially control the lac operon. So there are the promoters which are IPTG inducibles are lac promoters, definitely. These promoters are present in plasmid like PUC series plasmid, PGEM series plasmid. Then there are two more promoters, the TAC promoter and the TRC promoter. They are very similar because they are tryptophan lac promoters. Both are tryptophan lac promoters actually. And in case of a promoter, there are two regions which are very important. -10 region, which usually has the Tata box, and the -35 region. These two regions are very important, the sequence here, for understanding how much the strength of a promoter is. So here, the -10 region is from a lac UV5 promoter, and -35 is of the TRP promoter. Here also it's the same, but the distance between -10 and -35 sites is different. So in example of tag promoters are in PKK 223 plasmid. In case of TRC promoter, it is PLRC 99A. There are certain bacteriophage promoters as well. So for example, lambda PL promoter and the T7 promoter. So these two promoters they have very tight control over the protein expression. Um, these are as well, these are inducible promoters as well. They can be induced by IPTG. What is essential because they have tight control, very toxic proteins can be, um, produced using these promoters, because if a protein is toxic to the cell and it is produced right from the very beginning, what happens is the cells will not be able to grow, leading to less amount of protein getting produced. But when we induce a cell, we induce it at a later stage of growth. What happens there is at, let's say, 0.8 OD, so there we sufficiently have number of cells, and they together will synthesize protein, which will cause definitely a little bit low growth or stunted growth of cells. But since there are many cells to begin with, the protein will be higher. We will get higher yield of protein. So in case of T7 promoter, for example, the E. coli strain in which you will transform this will have the T7 RNA polymerase gene. So it's again a engineered E. coli strain. Now, T7 RNA polymerase is under the strong control of lac UV5 promoter, and when you induce with IPTG, the lac UV5 promoter will cause overexpression of T7 RNA polymerase, leading to your protein of interest. So let's say if you're using a PET series vector, PET 28A, let's say it has a T7 promoter. So, and then it will have your gene of interest. This is the target gene. So it will cause RNA polymerase will bind to this T7 promoter and will overexpress your protein, whereas the other proteins in the cell will not really be overexpressed. So this is the idea behind it. Now, protein production essentially leads what are the steps? First is the transformation. So transformation is essentially where you insert the DNA into the cell, then you cause growth of bacteria. from OD 600, it ranges from zero point 0.6 to zero point 0.7. There we give IPTG or induction. IPTG induction. IPTG is usually given in the range of 0 to 1 millimolar. And it could be given from 3 to 6 hours to check whether your proteins are getting is produced. And then what you need to do is to recover uh, of bacteria and analysis of protein. So for that, there is a centrifugation step at 8,000 to 9,000 RPM. And then you usually go for an SDS page to understand what is happening, whether your protein is there or not. Now, there are certain factors which affect the protein synthesis in E. coli. One is the translation efficiency. Uh, so the composition of promoters, especially the Shine Dalgarno sequence, these all things can influence how much translation is happening. If there is a secondary structure in the promoter that can reduce the efficiency as well. What are the other factors? There could be growth conditions that could affect. So definitely media is one of the reasons. So which kind of media you are using uh, can affect protein production as well. So the carbon source in the media, the nitrogen source, these all are very important because they will provide the amino acids, or the nitrogen sources will actually provide the raw material for amino acids at least. Then there is the codon usage. Now, what is codon usage? So we know that codons are degenerate. So there are 61 codons for 20 amino acids. And there is a preference of a set of codons for a particular amino acid. That is called codon bias. So let's say you are taking codons, you are not performing codon optimization, you are just taking the gene as such, but the tRNA pool has insufficient codons. Is insufficient, basically. Is insufficient, then protein synthesis may stop, or it might also get delayed. So if it gets stopped, we will get truncated proteins. It may get delayed, leading to very less amount of protein getting produced. Then there are some fusion proteins which are there. So there are some fusion tags which can be attached in either N terminus or the C terminus. So we will discuss about these fusion tags and the vectors which have these tags and their features. So the fusion tag could be like beta galactosidase tag. The vectors which are present here is the PUC P blue script, PGEM series. They can be used for blue-white screening. And affinity purification. We'll talk about purification next week. There is another tag, that is maltose binding tag, also known as MBP. It is present in vector PMAL series vectors. They can be used for affinity purification. Then there is thioredoxin tag TRX. Again, PTRX vectors. Again, it can be used for affinity purification. Polyhistidine tag can be there. in PET series vector. GST tag can be there in PJEC series vector. PJEC series can be used for, again, affinity chromatography and reporter gene assay as well. Alkaline phosphatase, that is PTA1529. It can be used for reported gene assay. What are the advantages of fusion proteins? There is easy purification. Then another advantage is you can have targeted protein production. Compartments. For example, periplasmic targeting sequence, like the PEL-B sequence, if present, it will cause accumulation of protein at periplasm. And that helps in isolation, basically. Fusion proteins can also increase the half-life of protein because it hides the potential sites which can be used for protease um, protease attack that can be prevented. Another thing is the solubility. So these fusion tags can enhance the solubility of a protein, um, especially if present at the N terminus. Uh, right? So the fusion tags can be removed using reagents. These are chemical reagent, or they can be removed using a protease cutting site, which is inserted, let's say thrombin cleavage site inserted in between the tag and your protein. So reagents are like cyanogen bromide, hydroxylamine, enterokinase, factor 10, alpha thrombin, trypsin, sub listen. You can read about them. There could be protease tags as well, which can be attached uh, in between the tag and the target protein. Now, yeast can be used as an expression system because it's the simplest unicellular eukaryote, right? Yeast can be of two types. They could be non-methylotropic yeast. They cannot use one carbon compound as a energy source. They usually require either lactose or glucose, which are usually more than one carbon, like 6 carbon groups we are talking about, maltose, starch, complex carbon sources. The examples of this is Saccharomyces cerevisiae, K. lactis, or Yersinia lipolytica. These are examples. They are usually used for alcohol fermentation. The other kind of yeast are methylotropic yeast. They can use one carbon compound, let's say like methanol, as their energy source. Example would be Pichia pastoris or Pichia methanolica. So for using yeast as an expression system, again, we can transform them. Transformation is the process of inserting DNA into these cells. So we use either lithium acetate or electroporation for inserting the DNA. There are vectors, specific vectors for this, and there are anatrope markers or antibiotic resistant markers like hygromycin resistance and stuff like that for yeast. Promoters could be either constitutive like GAPDH or GAM1. These are constitutive promoter, or they could be inducible promoters of methanol oxidase and stuff like that. In the cytoplasm, the secretion is mostly high, but the recovery is difficult. So to produce protein in the yeast, the transform cell is grown in a 5 ml medium for 2 days at 28 degrees. And it is done with shaking at 180 RPM. The OD reaches around 5 to 7, something around in that range. And then we take it to a fresh media. Then there is induction. In this case, induction is done using methanol, 1% volume by volume, because these are methanotropic. Most probably they have methanol oxidase as a promoter, inducible promoter. And then we harvest the cells and do SDS page for identifying whether our protein is present or there. We can also give some secretory signals before our peptide protein sequence that will help this. Like there's an alpha mating signal in case of Saccharomyces cerevisiae. If this signal is present, then what happens is your protein will be secreted instead of being present in the cytoplasm. Insect cell lines are used. Mostly popular are baculovirus expression system. Here, the recovery of cytosolic protein is easier compared to yeast. So here what you have to do, you have to perform cloning as usual. Cloning is performed, following which there is recombinant baculovirus vector that is generated. Then screening as usual. Screening. Fourth would be culturing of recombinant insect cell lines, and 5 would be protein production. The vector DNA in this case has a upstream 5' sequence and a 3' downstream sequence. In between, there is a polymeric promoter, a cloning site, and a polyhedron termination site, right? And these upstream and downstream sequences are important for homologous recombination and essentially integration into the genome. It's very important. Mammalian expression system. Uh, Again, in this case, we use mammalian cell lines. We'll talk about these cell lines. Uh, it's very expensive. However, if you are trying to replicate protein synthesis in humans, these are the best way to do that. However, very expensive because media, the components, these are very expensive. There could be transient expression. Transient expression is where you go for transfection, and the protein synthesis is maximal till 72 hours post transfection. Or there could be permanent expression. That is, we have integration of foreign DNA to the genomic DNA. So we form specialized cell lines with such protein synthesis machinery. So some popular cell lines which we know are like HEK cells, HEK 293. They could be HeLa cells, CHO cell lines, 3T3. So HEK is from kidney. HeLa is from cervical cells. These are. CHO is ovary. Then there is 3T3, which is from fibroblast. Similarly, multiple cell lines are there. For transient expression, there is high expression for a short period. As I already told you, transfection is the preferred route. So how to perform that transient expression? We clone the gene in a vector, transform it into a bacteria, and we perform mini prep to isolate this plasmid. Following which we seed live cells. These are cell lines, mammalian cells essentially. Then we perform transfection. (24 to 48 or even 72 hours post transfection). Then we detect whether the protein has been synthesized using Western blotting. We'll talk about Western blotting in specific. In case of permanent expression, it's very similar to transient expression. But what is the difference here is that there is integration of foreign DNA to the host cell. host DNA. So there is a constitutive expression of this particular gene, particular protein. Then in case of mammalian expression, there could be again two types: constitutive expression using housekeeping genes, promoters of housekeeping gene. We talk about promoters. GAPDH, for example, these are non-inducible. Or there could be inducible expression. Uh, they are used for toxic proteins. The inducible expression system for mammalian cell lines is the TET on, TET off system, which is controlled by tetracycline. Then there is Western blotting. In Western blotting, first you have to prepare the sample. So these are the steps. So sample preparation, as you can already see, following which you perform gel electrophoresis, usually an SDS page in most cases. Then there is a transfer of the gel, I mean the protein from gel to membrane transfer, and usually pond shoe stain is used to visualize this transfer of protein. Then we wash away the stain. Then there is blocking. Blocking is performed with either 5% skimmed milk or 5% fat-free BSA. Then we go for probing. So probing is basically antibodies are given. There could be two-step probing where there is a primary antibody against your protein, and there is a secondary antibody which is against this primary antibody, and it is conjugated with uh, enzyme like horseradish peroxidase or alkaline phosphatase. We will talk about what are the probing steps. There could be one-step probing where the primary antibody itself has uh, it itself has the conjugated enzyme. So then we go for blot development, where we develop the blot. So there are certain chromogenic or luminescent methods to do this. So if we go for the chromogenic methods, there is HRP based. Reagents could be like 4-chloronaphthol. It gives a purple precipitate. Uh, there could be TMB, which gives a dark purple stain. For luminescent, if it is HRP based, there is luminol and hydrogen peroxide, which gives a blue light because of the luminol. When it gets oxidized, it gives a blue light. Now questions. Guanine specific cleavage in maximum Gilbert sequencing is done using formic acid, hydrazine, dimethyl sulfate, or piperidine. So the answer to this would be dimethyl sulfate. The chemical nucleotide sequencing method, just give me a moment. Nucleotide sequencing is maximum Gilbert sequencing. Difference between deoxynucleotide and dideoxynucleotide is essentially 3' hydroxyl group is missing. Enzyme used in maximum Gilbert sequencing for 32p labeling is polynucleotide kinase. Blue colonies in blue-white screening actually tells you empty plasmid vector is present in the cell. Set of virulence gene located in Agrobacterium tumefaciens is activated by actosyringe. If a culture starts with 50 cells, the number of cells present after 5 generations would be 1600. The antibiotics with their gene products, when they match it, it would be A4. Ampicillin would be beta-lactamase. Kanamycin would be neomycin phosphotransferase. Tetracycline, ribosomal protection. And then chloramphenicol would be acetyltransferase. Protein synthesis corresponds to the process of formation of amino acid from mRNA. Cyanogen bromide is used for cleavage of junctions. It cleaves after methionine residues. The portion of growth curve where rapid growth of bacteria is observed is log phase. Adenovirus are vectors of mammals. Here, arrange the following in the correct order. So, southern blotting is for DNA, DNA hybrid. Western blotting is for antigen antibody reaction. Northern blotting is for RNA DNA hybrid, and DNA fingerprinting is done using southern blotting. The technique most suitable for detecting the presence of a gene is obviously Western blotting. Gene product is it's if it mentioned cleavage of peptide chain is done by trypsin. Others cannot go for. These are amino acids usually. So with this, I think it's end of this week's session. We will be meeting again next week in the same time. So if it's okay with all of you, we can stop the session now. Thank you all for joining in.